Let's go to the part 2, immunity and immune system. Immunity and immune system. Okay, before that, let me check whether you are getting the screen or not. So, immunity and immune system. Immune system. So most of the times, repeated questions only you can see in this area. In biology, questions won't be repeated in general. But the repetition of the questions you can see, like graft rejection type questions are repeated again and again in this area. So that's you need to check it out through this particular. Then the system that gives immune system, the system that gives immunity to the body by recognizing, responding, and remembering the foreign antigens. Here I say again that we are going to the brief explanation of immune system. The complete detailed explanation of the immune system stepwise available in the black. You please watch that videos. This is the just revision type explanation. Okay, then let me continue. Include lymphoid organs, tissues, cells, and soluble molecules like antibodies. They play role in allergic reaction, autoimmune disease, and organ transplantation. As I repeat you that very small area, but most of the times are getting question in this from the this particular area, and that is repeated even. Okay, the organ where origin, maturation, and proliferation. That lymphocytes based on that lymphocytes occur. Two types of lymphocytes that we say based on that maturation, proliferation, that the T and B lymphocytes, that primary lymphoid organs and secondary lymphoid organs. So the story of T and B lymphocytes, we can learn from here. That production of the T and B lymphocytes occur in the primary lymphoid organ. Maturation occur in the primary lymphoid organ. Wherever the activation occurs, that organ is called as secondary lymphoid organ. Okay, so let's talk about this. Here, immature lymphocytes like T and B differentiate into antigen sensitive lymphocytes like the bone marrow and thymus. Bone marrow, the production of here the detailed explanation. If you come over the B lymphocytes and T lymphocytes, both are produced in bone marrow. Maturation of the B lymphocytes occur in bone marrow. Maturation of the T lymphocytes occur in thymus. Okay, then wherever the production and maturation occur, that organs are called primary lymphoid organ. The thymus is a large during birth, but gradually reduce in size. We say that thymus is a child gland and become a very small size in puberty. Okay, then let's come to the secondary lymphoid organs. Secondary lymphoid organs, the matured lymphocytes migrate to these organs. Then like maturation of T and B lymphocyte, after maturation, they'll come here and activate. So where activation occurs, let me repeat, where production and maturation occur, that is primary lymphoid organ. Where activation occurs, that is called secondary lymphoid organ. So like spleen, lymph nodes, tonsils, Peyer's patches, very regular question here. Peyer's patches of small intestine and appendix, lymphoid organs in small intestine, they'll ask in that manner. Secondary lymphoid organs are the sites for interaction of the lymphocytes with antigen, which then proliferates into effector cells and memory cells. Showing the organs here for you, the bone marrow and the thymus, which is near to the heart, ventral side of the heart. And secondary lymphoid organs like where activation occurs, that is spleen and lymph nodes. In the lymphatic system, in lymph vessels, when they're running in one certain places, that means mostly in the joints, that lymph vessel will bulge over. We call them as lymph nodes. And some more lymphoid organs, tonsils we say, appendix, and the Peyer's patches, small intestine, just we say that in that particular T and B lymphocyte will stay and wait for the microbial to come. Okay, spleen. What is spleen? You should recognize what is spleen. That is secondary lymphoid organ, like that. Okay, so bean-shaped organ contain lymphocytes and phagocytes. Removes worn-out RBCs and microorganisms from the blood. It's a reservoir of erythrocytes even. Lymph nodes. What is lymph node? Secondary lymphoid organ. Secondary lymphoid tissue. Small solid structures found in the lymphatic system. They trap the microorganisms or other antigens. Trapped antigens activate lymphocytes and cause the immune response. Next, MALT, mucosa associated lymphoid tissue. That means after the production and maturation in the primary lymphoid organ, the mature T and B lymphocyte will go and migrate into certain places, like as said earlier, spleen, then lymph node, and then some places of mucosa places of body, like mucosa of trachea, mucosa of intestine. Likewise, they reach. So when they reach there, we commonly call it as mucosa associated lymphoid tissue. That means that lymphoid tissue is associated with the mucosa layer in that way. Okay, located within the lining of respiratory and digestive and urinogenital tract, it constitutes 50%. This is the question. It constitutes 50% of lymphoid tissue. So that immunity, what is immunity? Basically, the overall ability of the immune system to fight against the disease causing organisms. We say immunity, it is of two types, innate and acquired immunity. When it comes to the immunity, innate immunity, the immunity at the time of birth, by birth itself, what do you get? That is called innate. What is non-specific type of defense? No memory cells here. Acquired immunity, immunity developed during lifetime. We say pathogen specific immunity. That will come after birth, immunity. This kind of immunity will come after birth, where when we expose to that particular microbe. Let's come in detail. Innate immunity. It provides different barriers to the entry of foreign agents into our body. Like they include physical barriers, physiological barriers, cellular barriers, and cytokine barriers. The physical barrier, like a skin, even it is called first line of defense. The skin, which prevent the entry of foreign bodies. The mucus coating of the lining of the respiratory and gastrointestinal and urinogenital tract, which they trap the microbes. Then the physiological barriers like acid in the stomach, saliva, tears, that they have lysozyme is the enzyme present in them, antibacterial enzyme which dissolves the bacterial cell wall, both saliva and tears. Okay, did you see this? Whatever we are saying, also these are said to be the first line of defense, like skin, saliva, tears, stomach, acid of stomach, and the good bacteria. What all these are said to be the first line of defense. Okay. Next, cytokine barriers. That what you say, cellular barriers. The phagocytes like WBC, particularly neutrophils, we have to talk about polymorphonuclear leukocyte, with the word more pronounced for neutrophils itself. The monocytes and natural killer lymphocytes, macrophages, modified monocytes, we say macrophages. Next, cytokine barriers. Cytokine barriers are of two types again, interleukins and interferons. Here in innate immunity, we talk about interferons. The virus infected cells secrete protein called interferon, released from virally infected cells, and which go to the healthy cell, which provide the information of the virus. The virus infected cells secrete proteins called interferon, which protect non-infected cell from further viral infection, just informing them to the healthy cell that virus is there, that is be alert in that way. Acquired immunity. Immunity who come after birth, we say acquired immunity. Pathogen specific immunity we call. It's characterized by memory. First, when we encounter with the pathogen, the low intensity primary response. Primary response is comparatively late even. Then, but during the primary response, the first response, we are going to develop the memory cells. That memory cells, when second encounter if happened with the same pathogen, that a high intensity secondary response, we say anamnestic response. Secondary response is called anamnestic response. Just remember this word, very important. Okay, so the primary and secondary immune responses are carried out with the third line of defense cells. We say B lymphocytes and T lymphocytes. The B lymphocyte produce antibodies which fight with pathogens. T lymphocyte help B cell in what way? T helper cells we are talking about. They help B cell to produce antibodies. T cytotoxic cell directly they kill. What do you call that kind of immunity? Cell mediated immunity. The resting helper T helper cell become activated helper T cell and they provide in the information of the particular pathogen either to the B cell for humoral immunity or either to the T cytotoxic cells to the cell mediated immunity. Okay, so how will it happens? In a brief, it was explained the B cell finds an antigen which matches its receptors. Then it waits until is activated by helper T cell. Then the B cell divides to produce plasma and memory cells. Then the plasma cell produce antibodies that attach to the current type of invader. Then number 5, eater cells prefer intruders marked with antibodies and eat loads of them. Neutrophils are talking about. If the same intruder invades again, but memory cells will remember that and we activate much faster. That's what you say, natural active acquired immunity. The structure of antibody. Each antibody has 4 polypeptide chains. A very important area to get in the form of question. The 2 small chains and 2 larger heavy chains. What do you say? H2, L2. Light chain, heavy chain. Then different types of antibodies are present here like IgA, IgG, IgM, IgE and IgD. Immunoglobulin. Antibody have basically the 2 portions, variable region and constant region. Let's talk about that variable region. This is the variable region, means changing region, where antigen come and bind with the to this particular area. That means this site will based upon the type of antigen. And this is the constant region. What do you say? This part, FC region, that fragment constant region, we say. Each antibody has 4 polypeptide chains, 2 small light chains, 2 larger heavy chains, what we talk about. And acquired immune response are of 2 types, humoral and cell mediated. Antibody mediated response, humoral response, we say. That T cytotoxic cells do the particular response, we say cell mediated immunity. Then antibodies are found in blood plasma. So we call it as humoral immune response. Humor means itself body fluid. T lymphocytes mediate, T cytotoxic cells mediate cell mediated immunity. The cell mediated immunity causes graft rejection. The body is able to differentiate self and non-self. See, here we have to get back again. This is the area. We got 2 times in examination, recent time, last 5 years itself. Cell mediated immunity causes graft rejection. Tissue matching and blood group matching are essential before undertaking any graft or transplant, and the patient has to take immune suppressant throughout the life. Okay, so let's talk about the acquired immunity is of 2 types, active acquired immunity, passive acquired immunity. Natural active acquired immunity, artificial active acquired immunity. Passive again, natural passive acquired immunity, artificial passive acquired immunity. Let's take one after another. First of all, we'll go with the active immunity. Here the antibodies are produced in a host body in response to antigens, living or dead microbes or other proteins. It's a slow process that it is produced by 2 ways. During natural infection, that microbe came their own. When it comes, we fight for first time. We said our response for the first time is late, but we produce memory cells. That's a second, we react faster. What do you say that as natural active acquired immunity? In that, what is natural? Entry of pathogen is natural. What is active? Our immune system is active. What you acquire? Memory cells we acquire. Okay, in the same way, artificial active acquired immunity. We say simply as vaccination. Then injecting the microbe deliberately during immunization. That means that killed or inactivated virus, microbes, whatever. When we inject into the body, so we develop a response against them and we develop memory cells against them. So that what do you call vaccination in general terms? Artificial. What is artificial? We are injecting manually. Active, our immune system active. Acquire. What do you acquire? Memory cells. That means without exposing to the parasite naturally, we are going to get this. Passive immunity. What is the meaning of passive? Immune system is not working properly. We say passive. That is common for a newborn baby after immediate birth. When it comes out from the mother after parturition, we can't expect immediately starting from the beginning. The baby should work, immune system should work actively. So that's why mother send the milk to the baby. The milk, mother's milk, we call colostrum, is having so many antibodies. Okay, that antibodies contain IgA. Regular question. That mother to placenta through the fetus, whatever the antibodies which can pass through, that is IgG. That is a different story. But colostrum we are talking about now. That colostrum IgA antibodies are present there. Okay, artificial passive immunity. Like that when in the snake bite, in the tetany condition, we are taking antibodies from outside through injection. What do you say? Artificial passive. What is natural? Let's come from the beginning. Natural entry of antibodies natural. Why antibodies are coming? Because immune system is passive. Do you acquire memory cells? No. Likewise, same. Artificial. What is artificial? Antibodies we are taking from outside, which are prepared in somewhere else. Passive. Why we are taking? Because our immune system is passive. Acquire. Do you acquire memory cell? No. That means in passive immunity, we are not fighting. That's how you don't get any memory against them. When disease comes, again you have to use the medicine. So memory of the immune system, we are talking about that the immunization is based on the memory of the immune system. Active immunization, what do you call technically? Artificial active acquired immunity. In this, a preparation of vaccine, antigenic proteins of pathogen are inactivated and is introduced into the body. The antibodies produced against these antigens neutralize the pathogenic agents during actual infection. The vaccines also generate memory B and T cells that recognize the pathogen quickly, like polio vaccine, hepatitis B vaccine, DPT vaccine. The vaccines are produced using DNA recombinant technology, like hepatitis B vaccine produced from the yeast. Okay, so then when it comes to the passive immunization, that it is a direct injection of the preformed antibodies. Preformed antibodies. You'll find this line in previous questions. Or antitoxin when quick immune response is required. Like means in a case of snake bite, that when the snake poison is roaming all over the blood, we can't produce immediately. So that's we should take antibodies from outside, which are prepared in the hearts. So that what do you call it as passive immunization. Okay, allergy is also playing a vital role in examination, the part of immune system. Exaggerated immune response of the immune system to certain antigens present in the environment. We say allergy. Then allergens like that substances which cause immune response, like that mites in the dust, pollen, animal dander, the fur of the animals. So this is the important point to be noted down. The antibodies produced in response to the allergens are of IgE type, IgE. Symptoms like sneezing, watery eyes, running nose, difficulty in breathing. We are not here to learn all of them in detail. Actual income detail, anaphylactic allergy, atopic allergy, arthritis allergy, serum sickness, delayed hypersensitivity, various things are there. But learning all that in detail, if you forget this particular issue, like which antibodies are responsible, these are the regular question will come. So that's a focus on the points where the question comes. IgE type allergy. Allergy is due to the release of chemicals like histamine, serotonin, and from mast cells. Inflammation, we use it to say. To determine the cause of allergy, the patient is exposed to or injected with very small doses of possible allergen, and the reactions will be studied over. Then only they go for the further treatment. Means some medicines are also allergic for some kind of people, like penicillin is not suitable for all. Drugs like antihistamine. Inflammation is a very common process actually. That means sometimes unusual swelling will get on the skin. That we call allergy. Then what we use, that is because of what? Mast cells released histamine, bradykinin, serotonin, prostaglandin. Because of that, it is happened actually. That over response of the immune system, exaggerated immune response. So, what kind of medicines to be taken? Antihistamine. You see any allergic condition, any swelling it comes, any inflammation. Swelling means inflammation. Color, dollar, rubor, tumor. Inflammation contain four properties. Color, the heat generation, dollar, pain, rubor, reddening, tumor, swelling. So these four properties we say inflammation. So in the cases, due to what? Due to histamine, bradykinin, serotonin, prostaglandin, which are released from the mast cells. So what is the medicine then? Antihistamine and adrenaline and steroids quickly reduce the symptoms of allergy. Modern lifestyle results in lowering of immunity and more sensitivity to allergens. As we know, asthma is a very common problem that due to allergy it will come. Okay, so autoimmunity. So here, due to genetic and other unknown reasons, the body attacks self cells. This results in autoimmune disease. This results in autoimmune disease. Okay, so one of the very regular example, rheumatoid arthritis is autoimmune disorder. We see that we attack on our own cells. We attack on our own cells. That's it. This is the issues of immune system. We say the complete immune system. Okay, but I do remember. I do make you to remember again